For our final video, we're going to be looking at different strategies that researchers have taken to look at gene-based therapies for Friedreich's ataxia. While none of these therapies are currently available for treatment of patients, there's been progress made on multiple different avenues to look at different things that might be helpful given what we've learned so far about how we go from the error in the DNA to the lack of the protein to the symptoms that patients have. The first of these topics we're going to go over is called adeno-associated virus, also AAV for short. All we really need to know about this is that there are different types of viruses that exist throughout the world, and many of them work by entering cells and leaving their own DNA in there, hijacking the cell's own systems in order to replicate the virus itself. Well, researchers did something smart and said that if the virus can hijack our systems, maybe we can hijack the viruses in order to use it for our own purposes. Let's take a look at what would normally happen in nature. Let's say this AAV, the virus comes in, and it's going to enter a normal cell in the human body and leave a copy of its own DNA in the cell's nucleus, which is the area that the cell stores its own DNA in order to keep those blueprints there in order to have the instructions to make all those proteins. Well, when the virus leaves its own DNA there, it's essentially hijacking the systems that the cell uses to use the DNA as a blueprint. So our own infrastructure in the cell is going to look at the virus DNA and say, hey, we'll follow out those instructions and make whatever you want. In that case, it can make more copies of the virus or other proteins that the virus wants to make. It essentially takes over what the cell would normally do and instead uses it for its own purposes. But what some very clever scientists figured out they could do is that they could alter the DNA that's in the virus shell in order to use it as a vector to insert what we want to into a cell instead. Remember that in Friedreich's ataxia, the problem is that you don't have a proper functioning copy of that FXN gene, so you're not making the right protein. Well, what if we take this virus here, and instead of having virus DNA in it, we create a normal copy of the FXN gene in it and allow the virus to do its job of bringing that copy of the gene into the cell and leaving a new FXN gene. In that case, even if our own copies of the FXN gene aren't working properly, the new one that we've just added to it will. And as a result, even though before we weren't making the proper frataxin protein that's going to do its job and prevent Friedreich's ataxia, now we've inserted this new copy of the gene, and it's just going to hang out in the cell's nucleus and do its job for us. In fact, they've already did a study as far back as 2014 where they added the FXN gene to this virus and transplanted it into mice that had basically an equivalent to Friedreich's ataxia, and they found that these mice did in fact go on to produce larger amounts of this frataxin protein. It was a good proof of concept in essence. And the strategy hasn't just been used for Friedreich's ataxia so far. It's actually been used for a disease called SMA, or spinal muscle atrophy. In fact, they've gone so far for that there's a medication already out in use for it. Having said all this, this isn't a system that's 100% perfect yet or particularly ready to use in Friedreich's ataxia. There are risks that are associated with it, and while research is continuing, we have to keep in mind things like you can have a reaction whenever you use a virus in this manner. Your body recognizes virus shells, and it's not uncommon for individuals to have a reaction to that. It's known for causing liver inflammation. There's also always a risk that what if we implant too much of that DNA and we end up making many copies of a protein, for example. It's known that if you give too much frataxin to somebody, can actually have negative side effects. We kind of want that Goldilocks zone, so it can be hard to dial in the exact amount of the DNA that you give to somebody. Finally, we have to consider cost. Unfortunately, a lot of these genetic treatments, while they have the potential to be highly efficacious and maybe even curative, they also tend to be really expensive because a lot of research has to be done and the equipment involved is not cheap. For example, the medication that's used for SMA costs over $2 million for a single use. Having said that, As far as we know currently, you only need to give a single use of it as opposed to taking a pill every day for the rest of your life. So clearly there's a lot of advantages and disadvantages to this type of system. We're going to be taking a look at another one right now. For our next subject, I want to talk a little bit about Crispr Cas9. This is something that you may have heard about in the general press. It got a Nobel Prize for its research and discovery not that long ago. And it has a lot of promise for future treatment of Friedreich's ataxia, and there's been some studies involved with it so far. But let's learn a little bit about what it is before we go into exactly how it might be used to help treat or cure Friedreich's ataxia. Okay, CRISPR stands for Clustered, Regularly Interspaced, Short, Palindromic Repeats. We don't need to worry about too much about what that actually means, and we're just going to call it CRISPR from now on because that's a mouthful. But essentially, it's a portion of a bacteria DNA that it uses as a record or a library for its immune defense against viruses. We often think of viruses going after people or other animals, but they can go after bacteria too. Bacteria are small, but viruses are even smaller, and certain types of viruses like to inject their DNA into a bacteria in order to hijack that bacteria system. Well, bacteria over millions of years have developed ways to defend themselves against this system. Using this library in their own DNA, they've created a defense system where they remember previous types of viruses that have attacked them, keep a record inside this repeating segment here, and then use proteins with this record in order to attack the virus DNA. Let's look at that a little bit more carefully. So this virus DNA is coming inside the bacteria. We have this Cas9 protein, which is this defensive protein. There's lots of different types of Cas proteins. We're really just going to focus on Cas9. What Cas9 does is it can use a record of previous attacks from viruses in order to have a matching segment in itself that will recognize new virus DNA that comes in. It says, aha, I've seen your type before, I'm going to go up and basically chew you up. Why that's useful for us is because Cas9 protein has a way in order to snip DNA, which we'll see is going to be important for how we can use that to affect faulty genes like an error in Friedreich's ataxia, so that we might be able to make changes in that. Next, we're going to go over how scientists have affected CRISPR Cas9 in order to use it for our own purposes. Okay, so now we know about this CRISPR Cas9 system a little bit. How does it actually help us, particularly in regards to Friedreich's ataxia, where we know that the main problem is that in that FXN gene, we have too long of a repeat of this GAA over and over again in the DNA's code. Well, we know that CRISPR and Cas9 is a system that allows Cas9 to have a guide segment to identify specific portions of a DNA segment. Well, if we know exactly what we're looking for with our FXN gene, we can modify the Cas9 protein to have this guide segment locate to the specific part of our DNA sequence that we want to. All right, so let's say that this DNA sequence right here is in fact our FXN gene, and we've created a guide segment that can go and locate to exactly where we want it to. All right, now Cas9 is going to go down. We already talked about how it's good at making cuts in DNA, and it makes a cut. All right, normally when your DNA has a cut like this, your body and your cell recognizes that that's a problem, and it tries to rejoin those parts together. And that can cause a mutation sometimes, or it can just come back to normal function. It depends. With CRISPR Cas9, however, we could decide, hey, we've already made a cut here. Why not attach a new piece of information to the system and have some new DNA we want to insert into this area? That's if we want to add some function to this system. There are certain types of genetic diseases, for example, where instead of having too long of a system, like in Friedreich's ataxia, we're missing some crucial information, which is why you might want to add the part of the blueprint and that new DNA into the system that you want. However, in Friedreich's ataxia, what we really want to do is shorten these GAA repeats again and again. And so instead of just making a single cut and then having it repair itself from the normal body system, we can use this system to cut out a whole area of this portion of the DNA that we don't want. So Friedreich's ataxia, as we recall, is when that GAA repeat is, instead of only a couple times happening, it's happening 90 to over 1,000 times. We try and cut out big pieces of that instead in order to shorten it and return our FXN gene to a more normal function so we can create more of that frataxin protein. In fact, there's already been some studies in mice showing a proof of concept of this, where they had mice with really long segments of these GAA proteins, and they were able to cut out large segments of it in order to improve the amount of frataxin production. However, there's one more problem with this. When we talked about the AAV, that virus before that injected DNA into cells, that was relatively easy to get that DNA into the place we want. However, in this case, if you have a grown adult, it might be hard to get all these changes into all the billions of cells that a person has. All right, so we're going to have to talk about how we deliver this mechanism to the right cell. We're going to talk about that right now. Now that we've taken a look at how CRISPR can be used to modify DNA, we're going to understand how we can actually get it to the place it needs to be and what's already been done and what progress has been made in regards to Friedreich's ataxia. So to start off with, we have to understand that the body is made up of billions of different cells, from the neurons in our brain to the skin cells in our skin to all sorts of things. And it can be a little bit difficult to implement CRISPR Cas9 on all of these different cells in order to change that blueprint DNA code so that we can make the protein that we need so that we can avoid the symptoms of Friedreich's ataxia. But we've found some ways around this, at least in theory so far, and we have had some promising studies in that effect. To look at how we do this, we're going to talk a little bit about hematopoietic stem and progenitor cells. These are cells that we have throughout our body, but are particularly located within the marrow of our bones and are used as precursor cells that eventually become all types of different blood cells. These HSPCs can grow up and turn into different types of immune cells, red blood cells, platelets, what have you. Let's step back a bit. What happens if we alter these HSPCs so that we cut out that long GAA repeat? Well, then hypothetically, these HSPCs should have the correct blueprints and the correct DNA to make all the frataxin that we need. But remember, it's really more of our nerve cells and our heart cells and other things that are having the problem, so how does this help us if we fix these cells? Well, let's find out how. So we're gonna take a needle or some other method to get a bunch of these HSPCs and bring them to the lab. Once they're in the lab, we can just use it in a petri dish, or frankly, probably a more complicated machine than that, and use CRISPR in the lab on the HSPCs and cut out all these GAA repeats. Therefore, now our HSPCs are going to have a proper copy of that FXN gene so they can make all the frataxin they need. All right, what happens then? Well, if then you inject these HSPCs back into the body that they came from, all of a sudden you have normal progenitor cells in there that are creating all the frataxin that those cells need. But it's not enough yet because, as we just talked about, we need that frataxin in the right cells. Luckily, there are certain types of blood cells that are known to share. Specifically, these guys can grow up to become immune cells and keep going on until they become a type of immune cell called a macrophage. Macrophages actually connect to other types of cells in order to help them if those cells seem to be sick. In particular, they use a process called tunneling nanotubules, which is a pretty cool name, and they can use it to donate different things to other cells. So let's say this macrophage notices that a nerve cell of a certain type is not doing too great. It can then create this tubule to connect it and donate some of its own parts in order to help that cell. Specifically, that frataxin is in a part of the cell called the mitochondria, and it's thought that it can even ship little parts of itself over to that other cell, and then suddenly, even though our nerve cell still has the faulty copies of that FXN gene, it's got donated versions from the macrophage and should be able to function much better. So now we've spent some time looking at two of the main avenues that researchers have been looking into for a genetic treatment or potentially even a cure for Friedreich's ataxia. You may recall that I mentioned one of these studies was done as far back as 2014. There have been quite a few since and some before as well. So what does the timeline look like in terms of turning this research that seems to show at least some proof of concept and some promise into an actual treatment for people? Obviously, it's completely understandable that we want a treatment as soon as possible for what is a really devastating disease for many patients and families. What does that process of bringing this research to an actual person look like? Well, the testing that I've talked about so far has really mostly been done in lab and animal models, meaning that you can look at individual cells in a dish and see, oh, we were able to make changes, and now this cell that was having a problem before is able to make more frataxin. And likewise, you can look at a mouse, for example, that has a version of Friedreich's ataxia and see if you're able to make any changes there. It's a big jump to go from there to treating people. There have been several genetic treatments for other diseases produced so far, however, and there is a process to go through this. You may have heard of clinical trials before, maybe in terms of something like COVID, where they're talking about vaccine clinical trials, or any other number of things. Well, the Food and Drug Administration in America goes through 4 different phases of clinical trials for people. And to even get to this stage, you have to have done a lot of research ahead of time in order to think that, hey, we have something that's viably worked in the lab and in animals, and we're pretty confident that it could be usable in people, and really importantly, also safe. So first of all, they test a very small number of people first and check is our doses correct, and that's what's called a phase 1 clinical trial. You're not even necessarily looking for a response at that point in terms of improvement of symptoms. It's a little bit of an exception for genetic diseases, which we'll go through at the end. You then move on to phase 2 clinical trials where you're looking at, is this actually working at all? What are the major side effects in a relatively small group of people? Before going on to phase 3 and looking at really hopefully as many people as possible for its overall effectiveness, and if it seems to be safe, spread out across a whole group of people, and that the benefits outweigh the costs. At that point, if it seems to be effective, the FDA may consider approving it for the general population, and you might have follow-up studies later surveying whether it seems to be useful in the public or not. This system gets a little bit different in rare, especially genetic diseases like Friedreich's ataxia. Are you going to give a healthy volunteer without Friedreich's ataxia a genetic treatment that might alter their DNA as a phase 1 clinical trial? Probably not. Similarly, you don't necessarily have enough patients to do a large phase 2 or phase 3 clinical trial. But overall, we still use the system to a certain degree to decide whether or not we think a drug is even ready to be tested for patients in the first place. And these are the different concerns that we have to have when we decide whether or not we're going to even try it with patients. The first is, if this treatment is so expensive that it's impossible to realistically give it to patients, then it might not work out in the long run. We've seen that some of these genetic treatments can cost upwards of millions of dollars, and thankfully some of them have been approved by insurance, and given their potential for lifelong improvements in treatment and cure, it can definitely be worth it. But if a cure costs $100 million per person, it's going to be tough making that sell. Next, you have to be concerned, especially for something like a genetic treatment, that it can cause reactions in patients. If you have a major allergic reaction to it or some other issue, then the side effects might be too dangerous to even consider treating it. We talked especially last time with Crispr Cas9 about how you even deliver a treatment like this. And before you can consider clinical trials, we have to have something pinned down of how do we actually cause these genetic changes in the cells that we need to. And next, we have to consider whether or not the changes that we're making are hitting the right target. We definitely only want to affect that faulty FXN gene, and we want the greatest accuracy as possible with our genetic changes so that we don't cause unintended side effects, which could have disastrous complications for patients. But while there are currently no genetic therapies available for Friedreich's ataxia, there are reasons to be optimistic. Until 2017, there were no approved gene therapies for any type of condition, despite the fact that there had been research into gene therapies for a variety of different cancers and inherited conditions since the 1990s. Fact is that in the last decade, there's been an explosion in terms of funding and available technologies to research these types of conditions. As of my last count of last year, there were 8 approved genetic therapies for different types of conditions between the United States and the European Union, along with 13 others in phase 3 trials and dozens in phase 1 and phase 2 trials. Thank you for watching. I hope this series of videos has been informative and helpful, and like me, I hope that you're looking forward to any further advances that are made in terms of treatment and hopefully a cure for Friedreich's ataxia.